Sorry, continue.
So I just want to start off my talk by thanking University of Queensland and the Venom Evolution Lab in particular for giving me a position as a PhD student where I've done all of this research. The Australian government's helped fund my tuition, scholarship, etc. And of course, the UQ Herpetological Society for having me as a speaker and having you all as an audience. It's going to be great. So our story kind of starts on December 7th, 1941, a day that will live in infamy. This is a picture of the attack on Pearl Harbor, but on the same day, the Japanese launched a number of surprise attacks around the Pacific, and this is what started the entire Pacific theater of World War II. The Japanese very quickly they weren't met with much resistance and very quickly took over a lot of territory, including Thailand, Indonesia, and most of New Guinea, which I'm sure is somewhat familiar to the Australians in the audience because they used bases in these places to then launch attacks actually on the Australian mainland, including Darwin and Townsville. And it wasn't until a couple years later that the Allies actually kind of got their act together and started a counteroffensive and started retaking territory. And one of the first places that was retaken by the Allies was Manus Island, for reasons that are entirely unrelated to its notoriety today. So it was recaptured in March 1944, which is almost like 2 years after the bombing of Darwin. And it played an important role in the rest of the war, largely because of this bay here. This is called Seattle Harbor. It's a very sheltered spot, and it's a lot of sheltered water. So they set up a bunch of wharves, dry docks, airfields. There's just a huge amount of infrastructure. And then after the end of the war, this spot was used as a staging ground for the salvage operations because throughout the war, obviously lots of military installations had gone up throughout the South Pacific. Lots of various pieces of material had been damaged or abandoned for whatever reason. Lots of that was gathered up and brought to Manus Island after the war was done, and those were no longer needed. From Manus Island, they were brought to Guam. Guam is, to this day, probably the most important U.S. airbase west of Hawaii, so the U.S. military knew that they would continue to be operating on Guam, which is why it's the location for all this salvage material to be brought there. Before the events that we're talking about here today, Guam had lots of native birds and no native snakes. It had 18 native species of birds, 2 of which were endemic and found only on Guam, including this Guam rail, as well as a number of introduced species and some migrants, so very large bird diversity. Then there started to be rumors of snakes. And the first published evidence of this comes from 1953, where someone had written that they'd heard rumors for the past few years that one could sometimes see snakes around the main military harbor in Guam. 2 years later, in the governor's official annual report, they confirmed that they had captured 8 snakes in the 4 years previous, including some individuals that were well over 2 meters, so obviously very healthy adults. And these were identified as Boiga irregularis, which is the brown tree snake. This is actually the same species of brown tree snake that we have here in Brisbane. And they're a member of the of the genus Boiga, which is found across Asia from like India and Pakistan area, all the way here to Australia. They deliver venom to their prey through grooved fangs in the back of their mouth. And it's kind of important to note that Boiga irregularis, the brown tree snake, is not really a particularly outstanding member of this genus. It obviously gets very large. They were finding specimens, like I said, well over 2 meters in Guam. You'll see a picture soon enough of a quite large individual. But they're not the biggest Boiga species. They're very good climbers, but they're not necessarily the best climber out of the Boiga. They have a very broad diet where when they're young, they'll tend to eat small lizards, but as they grow older, they will start to take eggs, various fledgling birds, even adult birds, depending on the size, and the large ones will even take small mammals and rodents. So very broad diet, which is obviously good as an invader, but again, not something that's unique to Boiga irregularis among the genus. And everywhere they're found, it's important to keep in mind that these snakes are able to coexist with local bird populations. We don't have forests devoid of birds out here in Brisbane just because we have Boiga. But on Guam, as soon as the snake established itself and moved across the island, the bird populations, each of these graphs represents a population over time, almost all of them just fell off a cliff. Currently, there's only 3 species of birds really left on Guam. Of the 2 that were endemic to Guam, the flycatcher is entirely extinct, and the Guam rail is only alive in captivity today. The forests of Guam these days are silent and full of spider webs due to the lack of insectivores. And while they've gotten a lot less study, we're reasonably certain that the brown tree snake has similar effects on the populations of local bats and some of the local lizard species, though many small lizards tend to have high reproductive rates, and some of them have kept up with the snake. Beyond their ecological impacts, though, these snakes are just nuisances to the people on the island. They pull stunts like this, except on the power lines, and cause electrical failures across the island. There have been a couple hundred snake-caused power outages in the past couple decades. And they can enter homes. This is the large individual I was telling you to see. That's a you don't see brown tree snakes like that here in Queensland. And so people don't like having snakes in their home, but on top of that, their bites can be kind of nasty. They are rear-fanged, like I mentioned, so they kind of have to open their mouth real wide and chew the fangs in to deliver much venom. But if they do, the results can be pretty unpleasant. You get tissue effects like the swelling and blistering here, but what's more dangerous is the neurotoxicity. In adults, there's dizziness, numbness, loss of balance, but small children who have received severe bites on Guam have been hospitalized for breathing problems that come from the neurotoxicity. So there's a little bit of there's human-snake conflict on top of the ecological destruction. And this is maybe a little counterintuitive. We're used to thinking of dangerous snakes as the front-fang snakes, kind of like the elapids, such as the eastern brown snake here, or vipers like this beautiful pit viper. But as researchers over the past few decades have really come to realize that many, many snakes are venomous. And the key aspect of this realization is that these venoms are basically, they exist for predation for the snakes. And since no snake eats humans, we're largely irrelevant to the evolutionary context of these venoms. So venoms that are perfectly effective for a snake's predatory habits, range from totally harmless to humans all the way through extremely dangerous. And similarly, their venom delivery mechanisms range from barely differentiated teeth in the back of the mouth up through, hinged, hollow fangs, very specialized adaptation like you find in vipers. And the genus Boiga actually played an important role in this, in the early research into the venom of these non-front-fanged snakes. So for instance, an important toxin that we're going to be talking about a lot is called denmotoxin. It was isolated from Boiga dendrophylla, which is the mangrove snake, this beautiful yellow and black snake found through lots of Southeast Asia. And this was the first non-front-fang snake toxin to have the structure of the protein solved. And this is kind of what one of those structures looks like. This actually is denmotoxin. And basically, a protein is a long chain of amino acids that folds up in a particular way. And this is just a cartoon that's kind of showing you the backbone of that chain of amino acids and how it folds up. I've highlighted here the amino acid known as cysteine. Cysteine is a special amino acid because its side group contains sulfide. And these sulfides, when they're near to each other, can actually form bonds, like strong molecular bonds between the two amino acids. And those kind of act as links across different parts of the chain, which helps stabilize the folding structure of these proteins. Denmotoxin in particular was very interesting because it's a three-finger toxin. They get their names from these 3 loops or fingers, and classically, they were thought of as the elapid neurotoxins. If you think about something like a death adder or a coral snake from the Americas, those often have lots of three-finger toxins in their venom. That's what causes the paralysis and other nerve problems that people tend to associate with bites like that. So it's very interesting to start finding these three-finger toxins in the bites, in the venom of snakes, whose bites aren't very dangerous, and who before the 2000s, people weren't really sure if they were ready to call these snakes venomous or not. So these classic venom proteins showing up in their in the secretions that people got from them was kind of a very convincing case. The same research group, just a few years later, published the discovery of a toxin they called irregularis, which actually came from Boiga irregularis itself. This is an interesting toxin in that it's composed of two different three-finger toxins. So you can see these look a lot like denmotoxin earlier. They still have those 10 normal structural cysteines that I highlighted previously, but each of them has a unique extra cysteine, which rather than forming a cysteine-cysteine bond within the protein, is actually left free and binds to, oops, wrong button, binds to the other subunit. This forms a toxin complex, which we call a dimer, and by doing this, it's suddenly 10 times more potent than denmotoxin was. And one of the interesting features of both of these, and these toxins, and because this is 10 times more toxic, it's even more pronounced with irregularis, is that these toxins were very specific to act on birds and reptiles rather than mammals. So that's another reason why we kind of think of Boiga as being not all that dangerous a bite, but can still be such just an efficient bird predator. They're many times more powerful when the target is a bird than when it's a mammal like us. So you've got an interesting toxin like this. It's in a snake that's done something interesting on the islands. It's highly toxic to birds, which this snake is notable for having driven to extinction. So the obvious next question is irregularis kind of the secret to the invasion of Guam? Is this why this invasion was so devastating? And that's kind of what our research was trying to get at. So we sequenced a bunch of venom gland transcriptomes to get at this, and the reason we went about it this way is because genomes are big, they've got very complicated structures, and the genome is basically the same in every tissue of an animal, whether it's in the eyeball or the toe, it's the same genome. The transcriptome, on the other hand, is just the messenger RNA that's actively being transcribed in that tissue to then get turned into functional proteins. So not only is the structure of those much similar because a lot of the non-coding regions of the genome are edited out, but the organism basically enriches the transcriptome for your genes of interest if you go to the right tissue. So if we look at the venom gland, we basically find venom proteins, or we certainly find more venom proteins than we do elsewhere. So it makes our job as researchers a lot easier to go look at transcriptomes than whole genomes, and it's much easier than trying to sequence like single genes because we don't really know what we're looking for. Venoms are very complex, and these non-front fang snake venoms have very little research done on them, so sometimes we're finding new toxin families when we go and look at transcriptomes. So we wanted a good comparison of these transcriptomes across Boiga. So we looked at two populations of Boiga irregularis from Indonesia and from here in Brisbane, two subspecies of Boiga dendrophylla, again, that mangrove snake. One Boiga cynodon, the dog-tooth snake, doing a very fun little defensive display here. We could sequence one from Boiga nigriceps, which is the black-headed cat snake, and one from Boiga trigonata, the gamma snake, which is found all the way over in like Pakistan, India. So this sampling covers basically the whole geographic range and the whole family tree of the Boiga genus. And within these data sets, I specifically pulled out all the three-finger toxins because that's kind of what we're looking for. We're interested in whether any of these other species are forming these dimer complexes. So then I took those sequences that we got and all the publicly available sequences that have been published before and made this huge mess of a family tree of these toxin sequences. Don't worry, you don't actually have to be able to read any of this. What I just want to point out is that there's this one branch of the tree which accounts for basically half of all the sequences. And within there we have both denmotoxin, like somewhere around here, and iridotoxin A down here, and iridotoxin B down there. So I've colored in the sequences that have those characteristic cysteines I talked about from the iridotoxin subunits. So the ones that have the cysteine, the extra cysteine in the same position as the iridotoxin A are colored in blue, and the ones with the cysteine in the same place as B are colored in red, and you'll probably be seeing those color schemes again. Just the interesting thing to note is that they all kind of pop up repeatedly across the tree, and there's some weird ones that have like both of them. So right away we can see that the picture is somewhat more complicated than we expected. I kind of confirmed some of these results using some independent analyses such as protein clustering and a protein similarity network. Again, they basically are what they sound like. The protein cluster is just an algorithmic way of saying these proteins are more like these proteins, and then assigning them a number based on which proteins they're like. And the cluster is this nice graphical representation that actually puts them close together. And so these are just kind of independent methods for confirming our results there. You can barely read the clusters, which are the numbers on these things. But once again, you can see that these denmotoxin-like sequences, which I call them that because denmotoxin is sitting in the middle of all of these, that they actually are like pretty distinct from all the rest of the toxins. And that once again, it's hard to distinguish this muddle of the A's and the B's and the ones that don't have either. So I keep referring to these as sequences, and I'll kind of explain that here. I mentioned earlier that proteins are this string of amino acids, even though they fold up into a three-dimensional shape. So if you stretch that string of proteins out into a line and then gave each amino acid its own letter, you could write it down as a string of letters, and that's what we have here. And that's what we call sequences. They're just a sequence of letters. And then I lined them all up into what we call an alignment. It's a very technical term. And I've highlighted the columns that represent those 10 structural cysteines, as well as the extra cysteine for iridotoxin A and iridotoxin B. And so again, and then there's just like a simplified version of that same family tree from earlier. So once again, you can see that there's multiple gains or losses. It's hard to tell if it's a gain or a loss evolutionarily just by reconstructing things, but you've got both popping up in a kind of strange pattern. And we've got sequences with both the A and the B cysteine present in Boiga irregularis, which is good. That's the species we first found this toxin in, so we would hope to find both of those there. But also in Boiga dendrophylla, the mangrove snake, Boiga nigriceps, the black-headed cat snake, and also the genus Trimorphodon, which is a North American genus. And there's something weird going on there because its sequence that has the A cysteine is also missing a couple of these other ones. It's more closely related to some of the B ones, but that's just something strange that we haven't really gotten into yet, but maybe in the future. On top of that, we found sequences with just the B cysteine in Boiga cynodon and the genus Telescopus, which is a very close relative of Boiga from Africa, but neither of those species had the A cysteine. And then Boiga trigonata didn't have the A or the B. So we postulate from this that the toxins with the B cysteine may have evolved fairly early in the common ancestor of Boiga and Telescopus, and maybe even the common ancestor of Boiga and Trimorphodon. And then more recently, some of the Boiga species have evolved the matching A cysteine to form this dimer complex. And then Trimorphodon has done something strange on its own, somewhat independently. So that's kind of what we think has been going on. So we can then predict that these species, and maybe some of the others that weren't in our sample, Boiga dendrophylla and Boiga nigroceps at least, will actually produce these dimer complexes. Actual evidence of this. And that's where we go to what we call protein gels. So we make a gel in the lab. You can think of it as basically being a lot like Jell-O, where like the dessert, or Australians call it jelly, right?
Jelly.
Jelly, yeah, okay, great. So that dessert is basically formed of a bunch of strains of gelatin that are all interlocked, and so we use some lab chemicals to make a similar structure and then try and pass proteins through it. And larger proteins will get kind of tangled up in the mesh, smaller ones will have an easier time traveling through, so it separates things out by size nicely. And then we also ran these venoms under what we call normal and reducing conditions. And what the reducing conditions do is they break those cysteine-cysteine bonds that I keep going on about. And so that will kind of turn those proteins into more of a floppy tangle of spaghetti, but still the big ones travel slower than the small ones. And most importantly, it'll break the connection between any complexes. So rather than two proteins locked together moving like one big protein, they'll be split into two, and they'll both travel much faster. So we did this using venoms from Boiga dendrophylla, the mangrove snake, one of the snakes we hope that will have this dimer complex, and Toxicodryas blandingi, which is what's pictured here. It's the closest, Toxicodryas is the genus that is closest related to Boiga. It also lives in Africa. As you can see, it basically looks a lot like those other pictures that Boiga has been showing you. It's very similar body and lifestyle. Its ecological niche is about the same. And it's even doing a very similar threat to display to that cynodon picture I had up earlier. So a nice little outgroup to compare our Boiga to. So this is what a gel actually looks like when you separate the proteins out by size and then stain them. So in the middle, we've just got what we call a ladder. It's basically a bunch of proteins of known weights. So that's how we can put those molecular weights on there. And then on the left, we've got Toxicodryas, non-reduced, so totally normal conditions, and then reduced. And you can see that these two columns are pretty much the same. You know, there's little differences because gels are messy and inexact, but basically the same. But if we come over to Boiga dendrophylla, all of a sudden, there's a band in this column, which is unreduced, that isn't there in the reduced conditions. And this is between 15 and 20 kilodaltons, so that's basically exactly the size we'd expect for two three-finger toxins joined together. And when we reduce those, when we reduce the venom, we break the cysteine bonds, all of a sudden, that band disappears, and the bands down here that are about the size we'd expect for single three-finger toxins become much heavier. So this is the first evidence that anyone's ever made of these dimers being formed in species outside of Boiga irregularis. And our data from earlier indicates that this goes even beyond dendrophylla to at least Boiga nigroceps, probably a couple of the other more closely related species. Because there are lots of Boiga species across their range, and we weren't able to test them all. But this does mean that iridotoxin is not unique to Boiga irregularis. So it's unlikely to explain why it was that snake that caused this devastating invasion on Guam. Now, it is still a very toxic bird toxin, so it may explain why the brown tree snake was as destructive as it was to the bird populations, but it doesn't really help us explain why it was the brown tree snake. We also found those iridotoxin-like cysteines in genera outside of Boiga, which is just a weird story and doesn't really get to the point of this, but we'll hopefully have a talk about that later. But that does leave us with the question of why did this invasion happen the way it did? And I think you really have to go back to the accidents of history, the contingent facts that led to this. And it all starts, much like this talk, with the military. They brought Boiga irregularis to Guam. They've done studies and found that the Boiga irregularis on Guam are most closely related to those from the Admiralty Islands, which includes Manus Island, where all these war material was stored in provided nice little snake homes before it was shipped to Guam. And then you get compounding problems after that. It was, it was brought to a military base. And military bases aren't known for being, open for visiting hours after dark, which is when these snakes are actually crawling around. So not many locals on base at that time. And all of the military personnel, almost all of them came from the U.S., especially the southern parts of the U.S., which there's lots of snakes there. And they've been, probably many of them had been involved in the Pacific campaign of World War II, going around much of the South Pacific and Southeast Asia. And again, lots of those places have healthy snake populations as well. So most of the soldiers there probably didn't even realize that a snake on Guam was something out of the ordinary if they did see one crawling around. And then finally, there's just the fact that ecological preservation is pretty low down the military's list of priorities, especially directly after World War II, going into the Vietnam War, etc. It's only somewhat recently that the military has actually started helping out with the anti-Boiga measures on Guam, though they're doing a very good job of that now. They definitely don't want them spreading to Hawaii. But so that explains how the brown tree snake got there and why it's the brown tree snake, and why it was given such a golden opportunity to set up a population. I was talking earlier about the first rumors of snakes are in the early 50s. Well, the war ended in 45, and then it's not identified as the brown tree snake until 55, so it's a decade after the war that people are actually starting to realize that this might be a problem, which is plenty of time for these snakes to set up a breeding population. And then you just get to the fact that Guam was pretty much the perfect place for a snake to come in and set up shop. It is very naive prey populations that hadn't had to deal with a snake predator for thousands, maybe millions of years. You've got this snake, Boiga is just the perfect snake to come and invade something if you want to kill all the birds, because it's such a great climber. They're very food motivated. The researchers on Guam have all sorts of funny stories of what these snakes get up to. They'll try and eat big wire cages that have been scented with birds in the traps. There's reports of them eating people's leftover spare ribs. Anything that smells remotely like food, these snakes will try and eat. And they start out, obviously, like I said earlier, by eating little lizards. And there's lots of those on Guam. Some of them native, some of them introduced. They've got things like the Asian house gecko like we've got around here. I think they've got some introduced skinks that are doing great too. So that's the food that gets all these baby snakes started, and they feed happily until they're big enough to start actually taking birds. And then they've also got other introduced prey, such as the rats that come with people or the chickens that come with people. And basically what this does is it sets up a predator-prey dynamic where instead of the predator population crashing after the prey population crashes, these snakes are able to maintain relatively high population levels because of these alternate food sources, even after they've decimated the populations of the local birds. So really what it comes down to, the invasion of Guam was just this perfect storm of a predator with all the right adaptations beforehand, a very, very forgiving ecosystem for it to invade. It was just a smorgasbord for these snakes. And then they were just given the perfect opportunity to set up a breeding population. And when you combine all those factors, you get extinctions. Any questions? Yes, Christina.
Was there any evidence that other snakes were seen on there, and it's just the Boiga irregularis was the one that took hold? Like other invading snakes that just didn't do as well, or is it really just been one? Is there any information about that?
So the only other invasive species of snake that I've heard about on Guam was, what is it, Leptotyphlops brahminus, the flower pot snake. It's basically a global distribution now because it's a little blind snake. It lives in soil. It's distributed with topsoil, flower pots.
We find it in St. Lucia.
Yeah, no, it's here. It's anywhere. It's literally everywhere in the world. Like, I mean, obviously not in cold regions, but no, I haven't heard of any other snakes trying to set up populations on Guam. Just because, like I said, nothing else was given such a golden opportunity of a lot of artificial habitat being brought to one place and then moved to another. Yeah.
What are the military doing to combat?
So there's a lot of measures in place. There's some trapping. The trapping it's hard to trap snakes, but they mostly done locally. So like around the harbor at the airport on Guam, they'll have funnel traps set up that are scented with bird. So they look a lot like fish or minnow traps, if people are familiar with those. They're basically a big tube, and then they've got cones going in on either side. So the snake crawls in and is hopefully too dumb to find the little hole, find its way back out. Or is too busy trying to eat like the wire mesh box in the middle that's got the bait in it. I hear they try and do that. Yeah?
Because they are so food motivated and fairly clumsy, what's the risk of like self-envenomation, and how does that affect the venom affect them? Is there any research on that?
It's a good question. Self-venom resistance is a complicated field that has not gotten a ton of study, especially not with Boiga. I'm sure they bite themselves plenty, knowing these snakes. Lots of snakes do seem to be resistant to their own venom, and snakes that specialize in eating venomous snakes are often also venom resistant, but it also does seem to vary on a case-by-case basis where some, like if a king cobra bites another king cobra, the one that gets bitten still usually dies. I don't know of any research on Boiga's resistance to its own toxins. Yeah.
I heard that mangrove monitors Varanus indicus was introduced to Guam maybe several years before for like controlling the population of mice. And did they play like a role in controlling the population number of Boiga irregularis?
So you're right. Yeah, there are introduced monitors. They don't seem to have taken off to the extent of the Boiga, and they obviously will eat snakes if given the chance. Monitors will eat just about anything, and most large predators will eat snakes because they're slow-moving tubes of delicious meat. But I don't think they're a significant predator to the snakes, and I don't think they were even like that significant of a predator to the birds on Guam too. There's been some testing of the hypothesis of like, could it have been other introduced predators at the same time? And the snakes moved fairly slowly across the island, and the declines in bird population are very well correlated with the expanding snake range. Yeah.
Are they now in every part of Guam?
Basically, there are parts of Guam that don't have great habitat. So like on the southern parts of the island, it gets more like savanna-ish. And so you find fewer ground tree snakes down there because there's fewer trees. But yeah, they're everywhere on the island that they want to be. Yeah.
I was just wondering if they've invaded any other islands, and also how many species of Boiga there are.
Species of Boiga, I forget off the top of my head. It's over 10.
Okay.
They're pretty diverse genus. They haven't really invaded other islands, which is kind of a nice natural experiment. You have a lot of neighboring islands that have similar bird populations, and for the most part, they're doing just fine. You like just go across a little channel of ocean, and all of a sudden there's no snakes and lots of birds. Or even on Guam, like those bird populations that did survive are mostly protected because of environmental factors. Like there's a cave roosting population, and the Boiga can't climb up in the cave. And there's some urban populations that nest on like the sides of buildings and are pretty safe there from snakes. So it's things like that have allowed those birds that did survive to survive. All right, I think I'm going to call it.
Great, thanks.
Thank you.